Thank you. Hi, everybody. So today we have a speaker who's really interested in hearing from folks. So if you want to ask questions during the talk, please go ahead and do so using the Q&A function. You can find that at the bottom of your Zoom window. And other than that, I'm going to turn it over to Adan.
Great. Good afternoon, everyone. Welcome to the Biology Department seminar. And I want to begin by saying that today's seminar guest is being hosted by the Biology Department and the NIH IMSD program here at UMass Boston, program that she graciously visited earlier today. So thank you, Dr. Long. And with that, it is my sincere pleasure to introduce Dr. Terri Long. She is an associate professor in the Department of Plant and Microbial Biology at North Carolina State University. Terri received her bachelor's from University of North Carolina in Chapel Hill and her PhD in molecular genetics at the University of Georgia, where she studied plant immunity. Her postdoc was in the laboratory of Philip Benfey at Duke University, which is where I got to know her work in root biology and abiotic stress. And Terri has done some beautiful work in trying to better understand iron deficiency in plants. And in thinking about this introduction, I was reminded of one of her earlier studies, her earlier publications on the topic of that she worked on in the Benfey lab, where she described iron stress signaling mutant that she named pPYE. And it was a mutation in the transcription factor that regulated growth in iron levels. But it was really an elegant study, but it also reminded me on how you can actually have fun studying plants. And more recently, Terri and I have been working and collaborating on various projects related to diversity, equity, and inclusion with our international plant biology community, which has been a pleasure. And with that, Terri, welcome to UMass Boston and look forward to your talk.
Thank you so much, Adan. Thank you for inviting me here today. It was great to talk with your students. It's a nice, lively group. And so hopefully I'll keep things live and talk to you about multi-stress resilience and how iron kind of plays at the intersection of different stress conditions. So iron deficiency induced anemia is the most prevalent nutritional disorder in the world. Approximately a third of the world's population suffers from iron deficiency, and this is particularly prevalent in developing countries. But even in developed countries, women and children are particularly susceptible, and at least an overall estimated 10% decrease in overall human productivity. And so why plants? Well, socioeconomic reasons and religious reasons are two of the main reasons why people obtain their iron predominantly from plants, although meat is considered a better source of iron. And so understanding how plants absorb iron, how they deal with iron deficient conditions, is some of the kind of the focuses of our work. Now, it's not, why is iron deficiency a problem in plants? It's not because of the amount of iron that's in the Earth's soil. So it's abundant. It's the fourth most abundant element in the Earth's crust. However, it's not readily available. So iron will often form ferric oxides that are unavailable and that are only available through the weathering of rocks. And so you can see here some iron deficiency chlorosis susceptible variety of soybean that doesn't have what's called the strategy 1 response that enables them to bring in iron, whereas you have some IDC tolerant varieties who have this classic strategy 1 response, which I'll tell you about in the next few slides. And so then it leads to this paling and chlorotic phenotype that you see here in soybean. But if you look here, this is the plant that I study, Arabidopsis thaliana. It's a small plant, and it's considered the model plant. It's like the Drosophila of the plant world. And so these plants are grown in the presence of an iron chelator, and they also exhibit this chlorosis phenotype. And so how do plants deal with this process? So before they get to this stage, and these plants have been on a chelator for a long, long time, their first few days, they're actually inducing what's called the strategy 1 response. And this is particularly true of dicots and non-gramineous monocots. So basically, in response to iron deficient conditions, they will activate a proton ATPase that pumps protons into the rhizosphere. This lowers the pH, causing iron to become more soluble. And then iron is reduced from ferric iron to ferrous iron by membrane localized iron reductases localized at the in the membrane of the epidermal cells of the root epidermis. And then ferrous iron is transported into the epidermis through a membrane localized transporter. And then iron travels symplastically and apoplastically until they reach the vasculature, the xylem. And then iron is transported through the xylem from the root into the shoot. And there it's released into the mesophyll cells and incorporated into either enzymes involved in photosynthesis or enzymes involved in respiration, or even stored in the vacuoles or cell wall. Iron can also be transported into the phloem, and there it moves into reproductive tissues. And so we're learning more and more about these initial processes and the actual assimilation of iron. And so what I'm going to tell you about are some stories that are related to the regulatory mechanisms involved in controlling these processes. So I'll tell you a little bit about some of how iron plays an important role in abiotic stress and biotic stress, and how it also plays a role in development with respect to protein movement and the function of individual cell types. So I'm going to tell you about several stories that relate to the regulatory processes that are involved in a variety of different conditions. And everything kind of starts with understanding what's happening first in these first few steps of iron deficiency. So basically, there's a well-known transporter that controls the activity, a transcription factor that controls the activity of these three genes. And the questions that I've been interested in trying to answer is, what are the other regulatory mechanisms that control iron homeostasis? Okay. And so when I first started working in this field, one of the main, that transcription factor that I was referring to is called FIT. And doing a reverse genetic screen, we identified two other key players in this process that we named, as Adam mentioned, we named one PYE and we named the other Brutus. And throughout the years, we've learned more and more about how PYE and Brutus play an important role in controlling overall homeostasis. So this is a paper that was written by Christian DeBose's lab that kind of highlights some of the key players that control upstream responses to iron deficiency and some of the downstream components. And you see here that Brutus and PYE, we know now that they play key roles in this process. So what happens when you have, what do Brutus and PYE do? So loss of function of PYE leads to decreased tolerance to iron deficiency, suggesting that it's a positive regulator of the iron deficiency response. Whereas Brutus seems to be a repressor of the iron deficiency response, because when you knock out Brutus, then you get increased tolerance to iron deficiency. So I'll tell you a little bit about some of the current work that we're doing on, some of the past and current work that we're doing on these two proteins that help you understand how they may play a role in not just iron, but other conditions. So first, a little bit more about PYE. So PYE is a BHLH transcription factor. And BHLHs often form heterodimers. So if you use your imagination, you can imagine that PYE is the green BHLH, and then there's a partner that it interacts with, forming a heterodimer. And these heterodimers will sit down on DNA and control the expression or the repression of their targets. And so to try to get an idea of what those targets would be, one of the first things that we did was do, back in the day, we did a lot of microarray analysis. People do RNA-seq now, but 10 years ago, we did microarray analysis, and we found that many genes that are active in response to iron deficiency in wild-type plants are repressed. And there are some that are actually elevated under iron deficiency in a PYE mutant. This suggests that all sorts of genes involved in iron homeostasis may be directly or indirectly targeted by PYE. And the key thing that I want you to notice is that many genes that are involved in iron homeostasis actually seem to go up in response to iron deficiency, suggesting that PYE might be repressing some key iron homeostasis genes. And so I'll point out to you that, as again, PYE is actually a homolog. PYE is actually a member of a large family of transcription factors. And so here in the colored sections, you can see some close PYE homologs. And they are actually broken up into different subgroups. So there are about approximately 130 some odd transcription factors and VLH transcription factors encoded by the Arabidopsis genome. And they're broken up into different 12 different groups. And within group 4, there are 4 different subgroups, A, B, C, D. And PYE falls within this subgroup B. But it actually, we found through a series of experiments that they actually also interact with the subgroup C shown here. So we did yeast to hybrid analysis, and what you're seeing here is also what's called BIFC, and this allows you to basically take the fluorescence tag, a part of a fluorescence tag, and fuse it to PYE, or gene of interest, and fuse the other part of the tag to what we think might be its interacting partner. And then if you see a signal, you see a fluorescent signal. And so this biopsy analysis indicated to us that PYE interacts with several PYE homologs, and this interaction takes place specifically in the nucleus. And so this made sense to us because PYE is a transcription factor, and it should be acting in the nucleus. One of the key PYE homologs that we were interested in was a protein called ILR3. And previous studies by Bonnie Bartel's lab had shown that ILR3 may be controlling these vacuolar iron transporters, which control iron bioavailability. And this actually affects the ability for IAA and auxin to be conjugated. All right, so auxin availability basically. So we were like, oh shoot, ILR3 is doing something with iron as well. Let's see if ILR3 PYE interaction, how important that is. And so one of the first things we did was we checked out ILR3 phenotypes under minus iron, and we can see that they show a decreased iron tolerance, iron deficiency tolerance as well. So we made a double mutant, and we looked at PYE and ILR3 double mutants, and we saw that the phenotype looked very similar between the double and the single mutants, suggesting that they're part of the same regulatory network. So they form binding partners, and they may control the same set of genes. But when we do co-expression analysis, we find that ILR3 is co-expressed with genes involved in nothing necessarily related to iron homeostasis, whereas PYE is co-expressed with many well-known iron homeostasis genes, right? And so that was really curious to us, and it made us ask, well, what's ILR3 doing outside of this role in iron homeostasis? And around that same time, another paper came out showing a function of ILR3 in controlling glucosinolate biosynthesis. So here's the pathway, one of the pathways for glucosinolate, the biochemical pathways for glucosinolate biosynthesis. There are several transcription factors that control the activity of enzymes involved in this process. And it turns out that ILR3 is one of those transcription factors that controls the expression of some of these genes. So this suggests that ILR3 is controlling these genes, and the repression, and it actually seems to be repressing the activity of glucosinolate genes. And this was really intriguing to us because PYE itself seems to be maybe playing some sort of role in glucosinolate, because a lot of glucosinolate biosynthesis genes are also misregulated in a PYE mutant. So the question was, what roles might they be playing? Might the interplay between PYE and ILR3 might be playing in glucosinolate synthesis? So we did some quick qPCR, and we found out that perhaps ILR3, that a lot of these key enzymes involved in glucosinolate synthesis may be more upregulated in an ILR3 mutant than in a PYE mutant, again suggesting that PYE is an activator, whereas ILR3 is a repressor of glucosinolates, even though they're binding partners. And we then teamed up with Dan Klebenstein to try to gain insight into how this interplay might be playing out in plants. And so he measures, he's a guru with glucosinolates. So he measures long chain and short chain glucosinolates. And what he found was that ILR3, as he had published, does seem to be impacting long chain glucosinolate biosynthesis. So here's a NOVA analysis showing how significant the impact of the indicated factors are on glucosinolates, long chain glucosinolates. And you can see that ILR3, either individually or in combination, seem to show this impact. All right. However, and we also see a tissue-specific effect, which we already knew, right? However, we noticed that the iron effect doesn't seem to be prevalent unless you have ILR3 in combination with PYE. So this suggests that perhaps some interplay between ILR3 and PYE under iron deficiency plays an important role in controlling glucosinolates. And you can kind of see that play out when you look at the difference in the amount of glucosinolates in the root under plus iron versus the roots under minus iron, and in the shoots under plus iron, and then the shoots under minus iron. So we see that compared to wild type, we see just in general an increase in glucosinolates, particularly in the root. And we can see this particularly this is more evident when we look at the single versus the double and the double mutant compared to wild type. And so we wanted to see how this might play out in terms of real world conditions. So what are glucosinolates involved in? Glucosinolates are involved in wounding response. So we identified a wounding pathogen called cyst nematodes that actually infects Arabidopsis in addition to soybeans. It's a really important pathogen here in North Carolina. So we teamed up with Eric Davis in plant pathology, and we asked, what happens when we take our mutants and we grow them in the presence of this nematode? Do we see a change in the amount of nematode formation? And before I explain that data to you, I just wanted to point out, so what they do is they burrow into the root and they form these large syncytial cells near the xylem, sucking up nutrients. And so we wanted to see, if you infect plants, do you see a change in ILR3 expression, for example? What role might ILR3 be doing? So we looked at ILR3 GUS fusions and under plus and minus sign, and we saw that indeed ILR3 does show an increase in expression, but we also see this increase even in the presence of iron, in the presence of the cyst. So this suggested that the ILR3 is doing something, perhaps inducing glucosinolate synthesis that may impact cyst nematode infection. So then when we applied the nematodes to wild type versus our single mutants and our double mutants, we saw that there was a decrease in the amount of infection in the infection in our single and double mutants, suggesting that ILR3, perhaps in combination with pPYE, is modulating the amount of iron, perhaps to control the amount of either other iron requiring elements under wounding conditions. Right? So maybe ILR3 might be controlling glucosinolate so that iron can be available for nicotinamide synthesis or other processes that are important for bringing in iron. So a classic example of how plants can modify iron bioavailability. So that tells you a little bit about one of our biotic stress stories that we looked at, how iron might be playing a role, and some of our regulators might be playing a role in that process. I'll shift gears and talk a little bit about some of the abiotic stress studies that we've become interested in. So again, I'll remind you that our work is kind of focused on these two proteins and kind of moving out from those two proteins. And the second one, I'll spend some time talking about pPYE, but Brutus has this opposite response to iron deficiency. And so unlike pPYE, which is a straightforward BHLH transcription factor, Brutus encodes a protein with several interesting domains. It has 3 hemerethrin domains, and according to homology, hemerethrin domains bind iron and form these oxygen pockets. Brutus also encodes these other domains, one of which is a E3 ubiquitin ligase. And E3 ubiquitin ligases have been shown to interact with these other E1 and E2 proteins. And in that interaction, they bond with something and they cause whatever that something is to be degraded. So we were really intrigued by the possibility that Brutus might be an iron binding E3 ligase that controls its interacting proteins by causing their degradation. So one of the first things we did was check to see, okay, does Brutus really bind iron at the hemerethrin domains? We purified the protein, and we saw that when we purified the protein and quantified the amount of iron and other nutrients associated with the purified proteins, we see that there's a fair amount of iron and zinc associated with the full-length protein. But when we remove all 3 of these hemerethrin domains from Brutus, we see that there's a decrease in the amount of iron and zinc associated with the protein. Moreover, when we in vitro transcribe and translate the protein in the presence of increasing amounts of iron, we see that the protein seems to be destabilized, right? So this suggests that iron binding to Brutus may control the stability of the protein. We can even see this play out in vivo, when we look at the full-length Brutus protein under minus iron versus the truncated version of the protein. So we can really see that protein being stabilized in the absence of the hemerethrin domains. However, when we remove the E3 domain, we don't see that change in stability. So this suggests that the hemerethrin domains bind iron and control Brutus's stability or instability, whereas the E3 domain may be doing something else not associated with protein stability. So what might that thing be? To try to gain insight into that, we were like, all right, well, we did a non-directed yeast 2 hybrid analysis. So we took cDNA from a root and we conducted a yeast 2 hybrid analysis to see what other proteins Brutus might be interacting with and might be facilitating the degradation of. And we identified several transcription factors, 2 of which turns out to be the same BHLH transcription factors that PYE interacts with. So this was really exciting to us and suggests that there may be some interplay between Brutus and PYE, even if it's indirect. Right? And so to try to gain insight into whether those interactions might lead to the degradation of these 2 PYE homologs, we first checked to see, okay, is Brutus really an E3 ligase? So we did an in vitro ubiquitination assay. We take Brutus, full-length Brutus, and we add it to E1 and E2. Again, these 2 key proteins involved in the ubiquitination process. And we saw the formation of these long chain, these polyubiquitin chains. And this was, okay, this suggests, yes, we do see that the Brutus is capable of forming ubiquitin chains. All right. We also conducted biopsy to see if Brutus interacts with these PYE homologs in the nucleus. And indeed, we see that Brutus interacts, and I'm just showing ILR3 here. Brutus does interact with ILR3. We do see this signal, and this signal is associated with the nuclei. And I forgot to mention previously that these red dots are actually nuclei that have a red fluorescent marker in them. So this interaction between Brutus and ILR3 is in the nucleus, just like the interaction between ILR3 and PYE is in the nucleus. So then we did something called cell-free. Oh, do you have a question? Please feel free to. So then we did cell-free degradation assays to see, okay, is Brutus really causing the degradation of these PYE homologs? So what we do here is we basically extract protein from wild-type plants, and then we add in our purified PYE homolog. In this particular case, again, we're showing ILR3. And we see that when you add ILR3 to a wild-type, you get very little ILR3, suggesting that wild-type Brutus is degrading ILR3. When you remove Brutus, we see an accumulation. So this suggested that, okay, further evidence that Brutus facilitates the degradation of ILR3. And so what you might be asking is, what is this right here? So this is the version of Brutus that has been, where the hemerethrin domains are deleted. And what you have here is, again, is a very stabilized version of Brutus that acts very much like wild-type Brutus because you get degradation of ILR3. And so I'll point you back over to this side of the slide here, where you see the truncated version of Brutus, and you see way more formation of ubiquitin chains. This suggests that this very stable version of Brutus acts as an E3 ligase, which is why we see lots of ubiquitin chains here. So we have 2 different domains of Brutus that are doing 2 different functions, one for stability, one for E3 ligase activity. But Brutus doesn't just interact with PYE homologs. It interacts with several other proteins. And we were intrigued by the fact that it seems to interact with AT-VAS, another transcription factor in a different family, and then another protein called Importin alpha-3. And I'll tell you why those were intriguing to us. And so this interaction seems to occur in the dynamics of the interaction between the 2 things is interesting. So Brutus interacts with VAS in the cytosol and the nucleus, but it only appears to act with Importin in the nucleus. Now, Importin is not a transcription factor, all right? It is actually thought to be involved in nuclear transport. It interacts with something and causes this nuclear transport. So when we remove the E3 domain, we found that we lose the interaction between Brutus and VAS. This was really interesting to us because when we do this same study with Importin, however, we find that Brutus remains, that the interaction between Importin and Brutus still takes place. So this suggests that the interaction between Brutus and VAS may facilitate VAS's degradation, because this E3 domain is important for the interaction, whereas the interaction between Brutus and Importin has a completely different function and mechanism because it doesn't rely on the E3 domain. So we were like, oh, what's VAS2? This is not a BHLH transcription factor. But a series of studies that come out showing that these particular proteins were actually involved in abiotic stress, right? So VAS2 seems to repress drought tolerance, and I apologize for the quality of these pictures. I basically lifted them straight from the paper, and this is the best that I can do. But you can see here that the VAS2 loss of function means more stress and more drought tolerance. So this suggests that VAS2 seems to be repressing drought tolerance, but VAS1 and 2, VAS2 along with its homolog. But the VAS2 along with its homolog VAS1 seems to be activating biotic stress resistance. So these are plants that have been exposed to fungal pathogens or fungal derivatives. So it seems to be showing opposing roles in abiotic versus biotic stress. How might it be doing that? Well, this same group also showed that when you have a nuclear localized version of VAS, you can actually see that in the presence of MG132, which inhibits the activity of the 26S proteasome, which is required for Brutus's, which is required for E3 ligase activity, we can see that MG132 plays an important, in the presence of it, you see their protein accumulates, but in the absence, you see it doesn't. So this suggests that an E3 ligase, which relies on the 26S proteasome, might be involved in controlling the degradation of VAS2 when it is in the nucleus. And so this same study suggests that, I'm trying to move my little thing here, and suggests that nuclear localized VAS might even be degraded by the 26S proteasome through an E3 ligase. And so we were like, oh shoot, there's an E3 ligase that might be degrading VAS that people have been speculating about, and we see Brutus interacting with VAS. Well, obviously Brutus is at E3 ligase, so we did some experiments to kind of show that. So here is another cell-free degradation assay. All right, so here is where we basically take in, we apply these indicated proteins from these indicated lines in the presence of MG132. And remember, MG132 inhibits E3 ligase, the 26S proteasome, which the E3 ligase activity is relying upon. And then we add in VAS, the purified protein VAS, and we can see that in the absence of MG132, we don't see VAS, but in the presence of it, we do. However, in our Brutus mutants, we see that the protein accumulates in the absence of MG132. This suggests that, again, Brutus is important for the degradation of VAS. Right? We see that when we have, So, and then when we complement it, we see a very similar phenotype as our wild type. However, I want to point out that when we have the hemorrhagic deletion version of Brutus, it acts very much like wild type as well. But when we remove the E3 domain, we see an accumulation of VAS2. Again, suggesting that these E3 domains is important for the degradation of whatever Brutus is interacting with. In this particular case, we're talking about VAS2. So these cell-free degradation assays provide evidence that Brutus facilitates the degradation of VAS. And when you lose Brutus function, you don't see that degradation and the protein will accumulate in the absence of MG132. Right? And so in the absence of 132 is critical because this is when you, absence of MG132 is when you can have a functional E3 ligase. So this degradation was really exciting to us. We wanted to see, is this physiologically relevant? So we took some plants and we applied either desiccation stress or cold stress, which VAS seemed to be playing an important role in. And we assayed for VAS2 accumulation under increasing drought stress or under increasing times of cold stress. And we can see that the VAS2 protein seems to be decreasing. At the same time, we used a GFP version of Brutus transformed into Brutus, and we could actually see that the protein actually accumulates in the presence of drought stress or cold stress. So while Brutus is accumulating under these stress conditions, the protein that it might be degrading seems to be going down under these same conditions. Further evidence that Brutus facilitates the degradation of VAS in vivo. And so then we asked the question, okay, is this really impacting plant biology though? So I mean, we speculated then that if Brutus facilitates the degradation of VAS, then if you overexpress Brutus, you would get the same phenotype as a loss of VAS function. So here we have our overexpression lines, and we grow them under drought stress, and we can see that similar to our VAS1-2 loss of function mutants, you see increased drought tolerance in our Brutus overexpression lines. Right, so you might be asking, well, what about Importin? How let's not forget about this other protein that Brutus seems to be interacting with. What you notice that regardless of the presence of a functional form of Brutus, Importin seems to always be present. This provides further evidence that Brutus' interaction with Importin is very different from the interaction with these transcription factors like VAS and pPYE and pPYE homologs. And this is really interesting to us because Importins play an important role, as I said before, in the localization of proteins inside of a cell. So Importin alpha A will bind to some cargo, in our particular case, Brutus. And when it binds to this cargo, it also then binds to an Importin B protein. And this interaction between these three proteins facilitates the movement of it into the protein into the nucleus through the nuclear pore. Right? So we suspect that the interaction between Brutus and Importin has nothing to do with its degradation, but more to do with the fact that Importin facilitates the movement of Brutus into the nucleus, which is why you don't see that the impact of Brutus doesn't seem to be degrading ILR3. Brutus doesn't seem to be degrading Importin. So it's a really interesting protein with a bunch of different functions, and its interaction definitely is taking place in the nucleus, and Importin plays an important role in that. And so this is a little model that from a review that I wrote with Janneke Balk, and that kind of shows some of the functions of Brutus, and there's also some Brutus homologs. And so we think that Brutus seems to be repressing VAS, which plays an important role in cold and drought stress. And it may be most likely doing this in the leaves because of this function in drought stress potentially. But it also seems to be repressing ILR3 and potentially an ILR3 pPYE homolog, which then plays a role in controlling iron mobilization. And it's striking that this is repression because because if you look at a pPYE mutant, you actually find that there's an increase in the amount of iron in the mutants. They just can't seem to utilize the iron. So we think that pPYE is repressing many genes involved in proper mobilization of iron, and which impacts overall iron bioavailability. At the same time, ILR3 itself seems to play an important role in controlling other downstream components that are regulated by a Brutus homolog, which has been the focus of study from Janneke's lab. From the intercellular perspective, Brutus seems to be facilitating, the degradation of Brutus seems to be controlled by the binding to the hemerethrin domains, and then Importin itself facilitates this movement into the nucleus, which then controls its ability to control ILR3 and other pPYE homologs. So this is a nice little model that I like to show that kind of highlights the different functions and what we know about Brutus's function at this stage. But we weren't just interested in cold stress and drought stress. We've also become interested in other multi-stress conditions. And so a graduate student in my lab, Imani Madison, her project recently has been focused on iron deficiency stress and heat stress. And then she has another project that I'll tell you about a little bit later on in the talk. And Imani was really interested in the fact that when you look at Brutus and pPYE luciferase lines, you see that there's an increase in expression under iron deficiency, which is what we knew. But when you look at them after you apply a short heat shock, they seem to go down in expression. And then when you combine the two stress conditions, the expression of Brutus and pPYE seems to be intermediate. And so this is really intriguing to us and suggested that maybe there might be some interplay between iron stress and heat stress. And so to try to gain insight to that, she teamed up with Eli Buckner, who was a graduate student in Cranus Williams' lab. Cranus is one of my good colleagues in the Department of Electrical and Computer Engineering in the College of Engineering here at NC State. And what they did was they began to really track carefully root growth over time in response to heat and cold heat and iron stress. And they saw that, again, there's this very intermediate phenotype in terms of the growth rate of the roots over time. So then they began to ask the question, can we create a pipeline, an automated image analysis pipeline to really look at aspects of root growth at really, really high resolution that hadn't been achieved before? And so that's what they began to do. They exposed plants to these stress conditions, and then they looked at the root using light sheet microscopy. And then he created an automated and image analysis pipeline that allowed you to track certain regions of the root and extract 3D information, and then filter the information so that you can actually identify what's called a sum of squares to be able to see overall what are the differences in the root growth. And they weren't just interested in root growth, they were also interested in the cell cycle, because they wanted to see if the cell cycle with respect to the root growth was impacted by these two stress conditions. And so the cell cycle marker that we used was cyclin B1 fused to GFP. And so this is the machinery that we grew the plants in. To do light sheet, you really have to, I worked with my colleague, Rasu Zani, had designed something called the magic chamber. This is a multi-sample imaging growth chamber. And so basically it's a little tiny set of tubes that go into this 3D printed holder. It's about the size of a quarter. And you take these little pieces of PVC piping, I think it's PVC piping. And then Imani spent a lot of time. You would put a seed into the little bitty pipe, you put them onto a plate, and you let the seedlings grow through these little pipes. And then she would carefully take the little pipes and put them into the magic chamber. And then you put them into the light sheet, into the microscope, and then you can track the growth of the root over time. And she and Eli have written a couple of methods papers to describe this work. And so this gives you an idea of what the images look like. So here you can see the root, and you can measure the changes in the root growth over time, and they would extract images every 20 minutes for 24 hours. And here you can see the GFP signal, and you can actually envision the dynamics of the GFP signal relative to the root growth over time. And so this automated imaging analysis pipeline, which he named Biovision Tracker, was able to look at things like how much the signal for GFP persisted over time. How much did the signal spread over time and space? What is the average intensity of the signal? Or, what are the ends of the signal? How much did the GFP signal how elongated that was within a certain region of the root? And with that information, we could do this under multiple conditions. So you see heat stress, and iron deficiency, and a combination of those things. And so with this information, then he was able to do, Imani and Eli were able to do a sum of squares to integrate all of that data over time. And indeed, what they found is that the impact on all these different parameters on under iron deficiency was very different from that, or almost the opposite of what they saw under heat stress. So this suggests, and then when you look at them in combination, you see intermediate response, just like what we saw with root growth. This provides further evidence that iron deficiency, their iron deficiency response with respect to growth and the cell cycle is antagonistic to heat stress. And this makes sense in terms of ferroptosis. And we think that iron deficiency sort of negates ferroptosis, which is what happens when you have too much iron and ROS stress. And so heat stress iron deficiency may negate the impact of heat stress, which also causes ROS accumulation. So this is why we think that these two conditions may be acting antagonistically. So the last few stories I'll tell you about talk a little bit about development, protein movement, and how what we learn about protein, what we know about protein movement, and how that might help us gain insight into different cell types. So as I told you, and I haven't pointed out to you at this point, that ILR3 and PYE both seem to be mobile proteins. They seem so for example, ILR3 and PYE are expressed in the vasculature, but they seem to move to other cell types if you look at the GFP signal. And so my previous graduate student was really intrigued by this, and she teamed up with Ross Sutani again to use something called scanning fluorescence correlation spectroscopy coupled with pair correlation function to kind of map out the movement of these proteins over time. And she could see that when you quantify this movement, the PYE seems to be acting like a mobile protein, which is the free GFP, versus an immobile protein. And this analysis also indicated that that movement was from the vasculature to the epidermis cells, from the outer inner cells to the outer cells. So then she made a bunch of different transgenic lines where PYE was expressed in individual cell types. And quite often she would find versions where PYE would be expressed in one cell type, for example, here in the epidermis, and it would stay in the epidermis. But she also found lines in which PYE would be expressed in the epidermis and it would move to the cortex, or be expressed in the cortex and move to the vasculature and endodermis. And so we were really intrigued by this movement, and maybe we wondered if we can utilize that movement to try to figure out what PYE might be doing. And so she took these lines and she looked at them to see if they would be in facilitating rhizosphere acidification. Right. So, for example, here is our wild type line, and when you expose wild type plants to iron deficient conditions, they'll acidify the rhizosphere. And you can see this using this bromocresol purple stain here. And this yellowing indicates acidification. PYE mutants don't acidify the rhizosphere. When you complement PYE mutants with the wild type version, you see the acidification. All right. But when you express PYE in the vasculature and it stays in the vasculature or it moves to the next cell type, the endodermis, we don't see this acidification. We also don't see this acidification when you just keep it in the epidermis. However, when it's in the cortex and it moves to the endodermis in the vasculature, when it's in the cortex and it moves more internally, or if it's in the epidermis and it moves into the cortex, when it moves into the more internal cells, we do see acidification. So this suggests that the cortex itself is important for PYE function. And we can also see this kind of playing out in the shoots as well. So here you have your PYE mutant looking very sad, a complemented line looking very nice and happy. But when you look at the VEN lines, again, you see that these plants are really suffering and your epidermis line is really suffering. But when you have it in the cortex, it seems to be complementing. This suggests that, again, what is going on with the cortex? And so we measure the amount of iron in these plants using iron and other metals using ICP mass spec. And I know this is a lot, but I want to just point out to you that the PYE on iron, this is iron plus conditions, and these are iron minus conditions. And PYE mutant always seems to have the same amount of iron and other metals as the CE-NV lines. These are the lines in which PYE is in the cortex, endodermis and vasculature. And this is true for iron, zinc, manganese, and cobalt. And this is true under iron deficiency as well. So again, PYE is very similar to the CE-NV lines. And this is strange because why is it that they have the same amount of metals? Why is it the PYE loss of function and the CE-NV line have similar amounts of metals, but they have such a different phenotype? PYE looks really sickly, but the CE-NV lines don't. And so to gain insights into this, we did RNA-seq, and I won't go into a lot of detail, but suffice it to say, we wanted to see how those overall transcriptional profiles compare between the different lines. And we found that using PCA analysis, the CE-NV lines look very similar to PYE again, whereas the VE line was very distinct, right? And then our wild type and our complemented lines look similar. So again, the CE-NV lines are similar transcriptionally to PYE, which is very counterintuitive, is very odd. And we can also kind of see this play out in our RNA-seq analysis when we looked at well-known iron homeostasis genes. Again, CE-NV, PYE very similar, VEN very different. To try to figure out what that is, we did clustering analysis of all the genes that were differentially expressed in the different cell types. And we found, again, very similar things between the different lines. But one of the clusters that really stood out that helped to distinguish the PYE and the CE-NV lines is this particular cluster right here. And this particular cluster goes up in CE-NV, but doesn't go up in PYE. One of the really things that distinguishes the PYE transcript between the CE-NV lines. And we found that this particular cluster was enriched in genes involved in photosynthesis and carbon metabolism. And so this was really striking to us and suggested that when the shoot receives a signal, it goes down of iron deficiency, because remember, iron is really important in the shoot. This signal is conveyed to PYE. PYE then cuts on in the vasculature, travels to other cell types, and facilitates changes in iron mobility, localization, and then this within the cortex triggers an increase in carbon metabolism in the cortex, because other studies have shown that cortex is really important for carbon metabolism. So we think that PYE's function in the cortex plays an important role in carbon metabolism and in instability to deal with low iron conditions. Adon, I have another story, but I feel like I'm running out of time. So I think I'm going to skip that story for now, because I know you wanted 10 minutes for questions, right? Yeah, so we'll skip that story for now. And then I will, this is a story from Imani's project. So hopefully Imani's not watching, he's not upset. I'm going to skip over it. And so just in conclusion, we've done some work showing that PYE and ILR3 may control wounding response by modulating glucosinolate accumulation. And these heat and iron deficiency stress may act antagonistically, but that also some of what we've learned indicates a role for iron in controlling localization of certain proteins, which control the function of specific cell types. And with that, I'd like to thank all of my funding. The main funding agency is the National Science Foundation. Of course, the people in the lab, past and present members, as well as my good collaborators here. One of the main ones I want to point out are Ross Husani and Kranos Williams. In the last 2 seconds, I wanted to point out some of the, again, with Adan, we spent a lot of time thinking about how to make our field more diverse and inclusive. And so I serve on the Inclusivity Scholars Program Committee with the National North American and Arabidopsis Steering Committee, where we have a travel award to bring students to attend the International Conference on Arabidopsis Research. So if you're interested in trying to go to this conference and to present your work, please submit an application before March 15th. We're looking for good candidates and applicants. And also, if you want to learn a little bit more about some of the work that we're doing to change and make our field more inclusive and equitable, please check out the Changing Cultures and Climates website. That's a part of Plantain now, and learn more about our work. All right, so if there's no other questions, I will end it there. And I'll end it there, and I'll accept any questions if there are any.
Great. Thank you very much, Terri. This is fabulous. Maybe what I could do is I could start with the first question, but others feel free to put your questions in the Q&A and we'll read them to Terri, or Terri, feel free to also read them as they come in. Okay. So I'll ask the first question, and I'm curious, since Brutus is an E3 ligase, right? What does auxin do to it? The tier 1 is also a E3 ligase, and it seems to be connecting to potentially an auxin signal. So I'm curious about the interplay between auxin perception and sensing iron in the environment, and whether auxin can disturb the function of Brutus.
So because I think of tier 1 as, because I'm in different E3 ligases, right? So I'm thinking that Brutus and tier 1 are not related at all. But you're thinking because, why? What are you thinking is the link there? I'm probably just missing it.
Auxin signaling could impact glucosinolate synthesis, right? It's upstream of glucosinolate signals, and clearly ILR3 is downstream of Brutus. So I'm curious to know, okay, maybe auxin is actually disrupting Brutus function to alter glucosinolate function via its interaction with.
Oh, that's brilliant, Adon. I had never even thought about that. Yeah, I had literally not thought about that. And I'm gonna tell you the reason why I had not thought about that. I don't know if this is being recorded, but maybe you want to take this recorded part off. But we were working on this, and when I was first working on Brutus as a postdoc, I was working in Philip Benfey's lab, and Philip was not, maybe he still is not, he likes to avoid hormones. And he'd be like, hormones are so baroque, and I'd be like, are they broke? And no, he means baroque as in very complicated. Like, he was like, let's just avoid the whole hormone. So I've taken up that same kind of like avoidance of hormones because it is so complex. I mean, no way are these things acting in isolation. You're right. The link between auxin and ILR3 Brutus is something that I never thought about exploring. Simply because I had so much fun and dealing so much with other aspects of Brutus's function. But you may be right. It may be that Brutus, there's some sort of feedback mechanism whereby Brutus affects auxin availability, which then impacts, I don't know. And that's a proposal right there.
And if I may, the other connection is the story you didn't tell, because we know that vascular development is driven by auxin.
Exactly. Yeah, no, and I apologize for running. I should have placed that story a little earlier up. Yeah, so Imani's working on a project where we have markers that mark different levels of development of phloem. And then she's done, we did RNA-seq, and we did we've developed a regulatory network that looks at the progression of phloem development under plus iron and minus iron. And from that, then she's pulled out several potential hubs that we're characterizing now. But the interesting thing is that iron deficiency definitely seems to impact phloem enucleation because, and the idea came from the fact that red blood cells undergo enucleation as well. And iron is required for that enucleation process to occur. So we're like, well, maybe iron deficiency also impacts enucleation of the phloem. And the phloem has to be enucleated. We have to remove the nucleus from the phloem so that photosynthesis can move from the shoot into the root. So I forgot your question, Adon. I just use it as an opportunity to talk about that last story because what was your initial question again?
Well, you answered it.
Okay, good.
Yeah, you did.
Okay.
Brooke?
Yeah, thank you so much for the talk. I love seeing functional genetics work in Arabidopsis. It's always so neat as someone who works at the like organismal level to see all that, like how it's detective work. But so as someone who works in an organismal level, I was most struck by this idea that you can have multiple stresses interacting. So you saw that with the heat and the iron deficiency, but also when you were looking at the role of, I forget the name of it, the VOXs?
VOS, yes. Right.
VOZs, yeah. And their role in biotic and abiotic stress. And it seems like whenever we find these sort of master regulators of stress response, they're always involved in like everything.
Exactly.
I guess the question I have is, you know, it's like same with oxygen. They're Baroque too, I guess. Where do you kind of delimit your research, or do you just kind of keep pushing into those other?
I keep pushing until the funding runs out. we do whatever. I'd love to be able to do a study with, say, maybe Jeff's lab, and to just take these VOS mutants and just do a full-on fungal analysis to see. It's just a matter of having time, energy, and money to do it. But yeah, I suspect that Brutus is involved in a lot of other stress conditions, and it's just a matter of how far do you want to go with it, molecularly as well as physiologically. We could do another yeast to hybrid analysis, find a whole different set of interactors and go down the rabbit hole that way too.
Maybe I can ask another question while we're waiting for any others from the audience. So the movement of pPYE is really amazing. So it almost is as if it's behaving like a sensor, a sort of carbon and iron sensor, which iron is definitely needed for photosynthesis, but it's functioning like a sensor of both how much carbon you have and how much iron you have in the system.
Exactly.
Is that where you're going with this, or what are your thoughts on this?
I had not thought of it as a sensor. I had thought of it as more like a signaler. Well, maybe that's the same thing. Maybe you're thinking, I think it's the same thing. Whereas it's like communicating the status of iron in the vasculature to the rest of the cell types. And just by chance, oh shoot, it's doing something to control bioavailability in all of these cells. But in the cortex, this is particularly important because the cortex, and again, other studies have shown, even work that I did when I was a postdoc, shows that the cortex is particularly enriched in genes involved in photosynthesis. And that's a really under-researched area, there's a lot of questions we have about it. What is the cortex doing? Why are so many genes involved in photosynthesis? Is this really contributing to the overall carbon status of the plant? We think it is. And so if you change the availability of iron in all the cells, including the cortex is going to really utilize that so that it can trigger photosynthesis, carbon metabolism in the shoot, in the root, potentially. And so we think it's doing the same thing in all the cells, but the cortex is particularly interesting because of the specific function of the cortex. And then that somehow contributes to plant tolerance, to plant tolerating iron deficiency better, or utilizing iron better. So hopefully the reviewers will think the same thing when we submit we submit this next week.
All right. I don't see any questions, so maybe this is a good time to end, and also mentioned, I'll put in the chat a link to our happy hour for those that might be interested. You're welcome to join us. We'll be taking a short break, maybe a 5-minute break, and then you can join us at the happy hour link that you see in the chat box. Terri, thank you very much for your visit, your virtual visit.
Thank you.
Enjoy the spring. It was wonderful to see you.
Wonderful to see you as well, Adon. Hopefully, I'll see you in the next
Oh,
meeting we have.
okay. It's not in the chat.
Oh, yeah,
Oh.
you said it to panelists.
Oh.
You gotta change the thing.
Okay.
But yes, please join us there. It's a more regular Zoom meeting with more casual interaction, so you can ask Dr.
Okay,
Long
let
any
me put
questions
it
you may have.
again.
Thank you, Brooke, for and Adon for being. And I know it's hard to sit up with your eyes open during the Zoom call. I appreciate your having some
No, you
live
kept it
feedback.
engaging. It was really interesting.
That's great. All right, so we'll take a break, and Terri, you're welcome to join us if you want.
Yeah, I can join
Okay.
for a few minutes. Yeah.
Okay. All right. Thank you, everyone.
Do I log off from this one, or do I?
Yes, you log off, and then there's the other one for the happy hour, the other Zoom link for the happy hour.
I see it now. Okay.
Yeah. All right.
Bye.
Feel free to take a break.
Okay. Thanks. You too. Bye.
Okay. Bye.